Dr. Miriam Bornhorst has a joint appointment at George Washington University Hospital and Children's National Hospital in Washington, D.C. Her main clinical interests are cancer predisposition, including neurofibromatosis, and neuro-oncology. She's an attending physician in the neuro-oncology clinic, medical director of the Cancer Genetics Program, and clinical director of the Gilbert Family Neurofibromatosis Clinic. Dr. Bornhorst is the CNH institution PI for 11 clinical trials, 8 of which are related to NF1. Her research focuses on early detection and treatment of patients at risk for developing cancer, ultimately improving long-term outcomes. We're very happy to have her. Let's welcome Dr. Bornhorst.
Thank you, everybody. And I'm actually going to go ahead and take my mask off, just to help me talk a little bit easier here. So I get to tell you today the story of Selumetinib and the FDA approval of Selumetinib. I am also going to talk a little bit about other Mech inhibitors and some future directions, but really going to focus a majority of this talk on how this medication became FDA approved and some of the data that came with the trials that were done for that. So I do have to, sorry, if I can get this to work. It was working earlier. Sorry, it was working earlier. It's on. Maybe it went to sleep. Maybe, that's okay. Yes. We're good. All right, good, perfect. I do have to disclose to you that I am a registry scientific advisor, on the scientific advisory board for the Selumetinib registry, but that really has no impact on the talk that I'm going to be giving today. So many of you have gone to talks this past few days and have probably heard many talks in the past about what NF1 is and how it works and how it impacts cells. But in order to really understand Mech inhibitors, including Selumetinib, and how they work, I just want to briefly go over this again to make sure that we're all sort of starting on the same page. So the NF1 gene is this tumor suppressor gene that's really important for regulating cell division. So this is a great picture here of a cell that's in the process of dividing. So one cell becomes 2 cells, and then those 2 cells become 2 additional cells, and then it kind of continues to divide. And there's times in your life when this is supposed to happen. So obviously when a baby's forming inside of a mom, the cells are supposed to be dividing in order to form all the different organs, the skin, and other different types of cells. But throughout your lifetime, there's also times that cells need to divide. You need to have your cells or your skin cells dividing to be able to, when you lose skin or if you damage your skin or just even for normal turnover, you have to have division of those cells. But there are also cells in your body that really shouldn't be dividing. And some of those are cells that are in the brain, also cells that are along the nerves in your body. So these cells at a certain developmental time point, once you've gotten a certain level of cells, or what we call the glial cells, which are the supporting cells for your neurons, they're not supposed to divide anymore. They're supposed to just stay where they are unless they have to divide, and that's where if you have damage or injury to that nerve. And so NF1 is actually a gene that is really, really important to maintain that sort of level of cells, especially around the neurons. And so what happens with NF1 is that if this gene is not working the way that it should be, you actually have division of cells in areas where they're not supposed to be dividing. So this is just sort of showing a picture here. So NF1 is caused by basically a spelling error, a mutation, or some sort of a change in the NF1 gene that affects its function. And so what you see on the top here is that when you have NF1, there's a change in one of your genes. So you can see I have the black circle there that shows that there's a spelling error in one of those genes that you're born with. And that makes it so that one of those genes is not working the way that it should, but you have a second one that's there and that gene is working the way that it's supposed to be. And so it, kind of fills in, I guess you could say, and you don't really see anything. And so you have normal function. But what happens if that second NF1 copy is affected, or you don't have any copies of the NF1 tumor suppressor gene doing the job that it's supposed to be doing? That's where we get the cell overgrowth. And so the tumor suppressor gene, so what it's supposed to do is tell these cells not to grow, but if there's nothing there to tell it not to grow, you're going to have cell growth. And that's where the tumors grow in NF1. And actually, we know now too that the cafe au lait spots that you get, or a lot of the other things with NF1, are something very similar. And so in your skin, if you have one copy that's lost and then you lose the other copy, that's where you get the cafe au lait spots. And that's why there's some thought that if you have skin damage or exposure to sun, things like that, that it can actually potentiate the cafe au laits to form. Where the second hit, quote unquote, or where you lose that tumor suppressor function happens in the body, tells you what type of tumor you're going to get. So if this happens in the optic nerve, you get something called an optic pathway glioma. So it's a tumor on the nerve that helps you see. If that happens on the skin, you can get cutaneous neurofibromas, or as I just mentioned, you can also get the cafe au lait spots and some of the other things that you can see on the skin. If this happens in your peripheral nerve, so basically any nerve and anywhere else in the rest of your body, you get a neurofibroma or a plexiform neurofibroma, which is going to be more of the focus of the talk today. So this is just showing what's happening inside the cell. So lots of different things going on inside the cell. And so at the top here, you get a signal, it hits the cell, and that signal is trying to tell the cell to divide. And so when you have your NF1 gene there and it's working the way that it's supposed to be, the pathways are not working, nothing's changing, and there's no change to the cell. It just sits there. That NF1 gene basically tells that cell, don't grow. Now is not a time for you to be changing, growing, or dividing. So that's how it's supposed to work. But what can happen sometimes is you'll get a signal that comes to the cell. Your NF protein is not doing the job it's supposed to be doing. And now these pathways that go to the nucleus and tell the cell to divide, like I was showing at the beginning, those pathways are active. And when they're active, then you get cell growth and you get the proliferation and a tumor. And so what people started looking at a while back is how can we change this, or how can we target this? And so if your NF1 protein's not working, for example, what can we do to bypass this so that we're sort of helping out the cells or keeping those cells from growing and dividing? And so NF1 is here, but you can see on here there's also something called RAS, there's something called RAF, and then it goes to Mech and ERK. And so a Mech inhibitor actually inhibits that Mech protein. And this works not only for NF1 when you have NF1 lost, but if you have a change in that RAS gene, it can work for that, and also that RAF gene, it can work for that. And so just to kind of show you, you've got your signal coming to the cell, you've got a Mech inhibitor now that's stopping that protein there, so when the signal tries to get through, it can't get through. It's no longer able to hit the nucleus and tell those cells to grow. So you have decreased cell growth, and you can even have sometimes death of these tumor cells because that signal is not able to get through. So at the very beginning, Mech inhibitors actually have been around for quite some time. They were actually first discovered in the late 1990s. It's interesting, if you read through the literature on Mech inhibitors, it was an accidental discovery. So people were trying to find something to help with inflammation and swelling. And so they developed this compound, and they thought that it was going to be the next steroid, I guess, or something that would help with inflammation, swelling in patients who needed that for example, rheumatoid arthritis or joint swelling. And it didn't work quite that way, but they did find that it inhibited this Mech protein, and because it inhibited this Mech protein, if you had a change in your NF1 gene, your RAF gene, or your RAS genes, that it inhibited that pathway and actually helped with tumors. And so it was a really great discovery that somebody made for actually for a lot of people. And so melanoma and non-small cell lung cancer are two types of cancers that also benefit from these. And the reason I'm bringing this up is because they actually were the first studies that were done with Mech inhibitors way back before we even thought about this for NF1. And so these drugs really have been around for quite some time. And you can see on here that trametinib was actually approved back in 2013. Cobimetinib, which is another Mech inhibitor, was approved back in 2015, and binimetinib was approved back in 2018, but for different indications, so the melanoma and non-small cell lung cancer. So what did we do for NF? We saw that this was working for these other cancers, and we thought that it could potentially help with NF. So how is it that we can get this from the concept or the thought to something that might actually work in patients? So there's lots of different clinical trials and clinical trial names, and so I want to just go over those briefly with you so you sort of understand what each of these trials mean. We always start with what's called preclinical trials or preclinical studies. And so these are studies that are done not in patients, but in something else, another model, whether it be a mouse, a rat. They have the NF1 pigs now. You can also use cell lines. This is sort of the proof of concept studies that you can do outside of a patient. So that's the preclinical studies. That always has to happen first. After preclinical studies, you can do something called a phase 0 study. And what a phase 0 study does is it actually looks to see how well this is affecting the target or how well the drug is getting to its target. So an example of a phase 0 study for NF is actually you take a Mech inhibitor, you take a medication, and then let's say we're looking at cutaneous neurofibromas. So you take it for 2 or 3 days, and then they take that neurofibroma off, and then they look at it and see, was the drug able to get there? How well did that work? And then you can have a phase 1 study. So once you know the drug is getting to where it needs to go, you can do phase 1. And a phase 1 study is also looking at how well does the drug get there, but it's also looking at the safety of the medication. Is it safe? And if it is safe, what dose is it safe at? So how can we find the right dose to use for additional studies? Once we get the dose we need and we know that it's safe, we move to a phase 2 trial. So this tells you how well the medication works and expands on the safety of it. So instead of just looking at 6 patients, for example, we're now expanding it out to 50, 60, 70 patients, because we want to make sure that we really understand the safety of it, but also how well is this working in patients. And then finally, you can go to what's called a phase 3 study, which is looking not at necessarily the safety of it and how well it works, because we've already semi-established that, but how does this work in comparison to what else is out there? So for NF1, the treatment for plexiform neurofibromas is surgery. And so a phase 3 trial, for example, would be if you take a Mech inhibitor and or if you have surgery, what works better? And that would be the phase 3 trial. So for the NF1 patients and for the Mech inhibitors, the preclinical studies that were done, there were 2 major studies that were done. One of them was on a compound PD0325901, which has now been renamed to Selumetinib. And then actually there was another one that was done with Selumetinib. And what they did is they took a mouse model of plexiform neurofibromas, they gave them the medication, and they looked to see what happened to the tumor. So similar to what we would do in a patient. And what they found is that when the mice were taking these medications, they had improvement in their tumors. And with both medications, they had improvement with their tumors. So that was enough evidence for us to bring it then to the patients. So we then did a phase 1 clinical trial. This started in 2011, which I must admit was before I was even really practicing. So that was a little while ago, not forever ago, but a little while ago. And with this phase 1 trial, we looked at patients between 3 to 18 years of age, and primarily looking at patients that had inoperable plexiform neurofibromas, so tumors that could not be taken out, but also caused morbidity, so caused some sort of a symptom, or there was something that the patient had that they were complaining of, whether it be pain, weakness, disfigurement, things like that. And with this, they did twice daily dosing, and they started at 20 milligrams per meter squared, and then they moved it up from 20 to 25 to 30 milligrams per meter squared. So that's that dose finding part of the phase 1. So how can we get the best dose? And then they did safety measurements. And what they went ahead and found is that regardless of the dose, actually a lot of or most of the patients had a response. And so the response rate was about 72%. And you can see on here, it's split into the 20, the 25, and the 30 milligram per meter squared. And all of those different dose levels had response. But the side effect was probably a little bit better on the lower side of it. So what they ended up doing was taking right in the middle the 25 milligram per meter squared as the recommended phase 2 dose or the recommended dose for the next studies. And the other thing that they actually found with the phase 1 trial is that the results that you get is really when you're on the medication, but when you come off the medication, the tumor can start to grow again. So that was something that actually came out very early in those studies, that you really almost need to keep the medication going to get the benefit from it. Because you see this patient here in that study where they were having a consistent growth in their tumor. They started on treatment. You can see a nice drop when they started on treatment in terms of the volume of the tumor. They had to have the medication held for a little bit, and it came right back up again. They then went back on medication medication and they started to have response again, but that really sort of showed us that these are not medications that you can start and stop. They are ones that you do need to continue on. After the phase 1 trial, it moved to the phase 2 trial. So this one started in 2016. And this is one where there were multiple or more institutions involved, but we also had more patients that were recruited. So we had 2 arms of this one. The first one that's up here is the symptomatic arm, and then there was an asymptomatic arm. So we recruited 50 patients to the symptomatic arm, and then we recruited 25 to the asymptomatic arm. And so obviously for the symptomatic arm, we again were 2 to 18 years of age. They had an operable plexiform neurofibroma, and they had to have a symptom very similar to that phase 1 trial. But the second one was patients that were within that 2 to 18 years of age that had a plexiform that as a clinician we felt could potentially cause a problem, and therefore they were not symptomatic but could potentially become symptomatic. This is looking more at how can it prevent symptoms versus how can it treat symptoms. The dose was 25 milligrams per meter squared. The safety evaluation, so patients came in initially every 2 weeks, then every month, and then it eventually spaced out to every 4 to 6 months. They did response evaluations, so we really had a lot of evaluations the poor patients had to go through. They had to have functional evaluations, they had to have quality of life evaluations, MRIs, echoes, eye exams. It was pretty intense for a lot of our patients. And then the primary endpoint was how well did this affect your plexiform neurofibroma volume? So we were looking for a greater than 20% decrease in volume. And then secondary endpoints, of course, were how did this affect patients in terms of their quality of life and function. And so this is just kind of a list of what we saw in terms of our patients on this study. There were, again, 50 patients that were enrolled. It has sort of the date range for that, the median patient age, males versus females on here, the median target tumor size. So these were fairly large tumors in this group. And then the progressive status. So there were a number of patients that did have a progression at baseline, but not everybody. And then, of course, the number of morbidities they had. So it was an average of about 3 morbidities that patients had when coming into this trial. And so this is showing the response from that. And I'm sure many of you have seen this because this has been published now for a while, and it's something that's been very exciting to the NF community. And it's very similar to the phase 1 trial. We found a partial response in about 74% of patients. So this is really, really impressive and amazing for us to see. And then a durable partial response, which means that not only did you get a response, but it was maintained for a long period of time, we saw in about 56% of our patients. So that is also something that's really important, because sometimes you can get a response, but if it goes away after 4 months, it doesn't really matter. It needs to stay there for a long period of time. The other thing that was really important from this study is not only did we get that response, but patients reported functional improvements. And so patients had improvement in their movement, improvement in their weakness, improvement in the way they felt, improvement in their fatigue. One of my patients that was on this study, his tumor didn't shrink much, but his mobility was amazing. I mean, he went from barely being able to move the leg to being able to do everything and anything he wanted to do. So I think that one of the great things about this study was that capture of the functional improvement, because it really was important for not only patients to see, but also when it went to the FDA for them to see how this impacted our patients. And then one other thing that they just showed with this too is it decreases the risk for tumor progression. And so this is showing right here sort of a progression-free survival curve. And so this blue curve here is what is the likelihood that your plexiform neurofibroma is going to grow or progress over a period of time? And then the red line was there is what did we see on the medication? And clearly a lot more patients progressed if they were not on treatment than patients progressed if they were on treatment. So it sort of helped maintain that's growth. So safety is also really important with this. So we saw the benefit, we saw the functional benefit, but is it safe? Was it something that was tolerable and that patients were able to take, and it was safe for patients to continue on? We did have, of course, some patients that had some side effects, and this is sort of showing what we saw there. So 14 patients did have to have dose reductions due to an adverse event or due to a side effect. And so it just shows you that not everybody needs that 25 milligram per meter squared dose. Sometimes you need something a little bit less. And then there were some patients that also discontinued therapy. There were some of them that discontinued because they ultimately did have progressive disease. Some of them discontinued just because they had stable disease and weren't getting the response they were hoping for. And then, of course, the adverse events, some discontinued because of that, and then just other reasons. So the side effects that we saw that resulted in discontinuation, really bad diarrhea, weight gain. Actually, one person had a lot of weight gain and came off from that. Paronychia, which is an infection around the nails, was another reason for coming off. Skin issues, again, skin ulcerations, and then there was one person who had come off because of an elevated creatinine. So this is what, after that study, came out as the most common and the less common side effects. And I think that, this is a really long list, and I don't want to read through the whole thing, but I think what's important here is that there are definitely things that you need to be monitored for when you're on these medications, because they can happen. And most of these are things that we can treat as clinicians, or you can stop the medication, come on a lower dose, and they go away. But the ones that I want to pay particular attention to are the eye toxicity. So there were a few patients that did have mild retinal detachment that went away as soon as you stopped the medication. And then also changes in heart function. And so sometimes the ejection fraction would go down, you go off of medication, and then that would improve again. So when your doctor, if you're on the Mech inhibitor and your doctor says you need an echo, you need an echo. I mean, there's a reason that we're monitoring your heart function as well as the eyes. And then this is just showing some pictures of examples of the rash, the paronychia. I mean, it's real. It happens, definitely. I mean, I don't want to say anything negative about my teenage males, but I definitely saw a lot more paronychia in my teenage males than some of my younger kids and my teenage females. And, I think a lot of that just has to do with how you take care of your nails. But then the skin rashes can happen to anybody. And so what I've discovered using these medications, especially with this bad acneiform rash, is that if you start treating for acne before you even start the medicine, you're less likely to have this happen. And so I oftentimes will start something like doxycycline or something oral before I even start the Mech inhibitor, because it can get to be pretty bad. So this is just the April 10th, 2020 statement from the FDA saying that this was approved. And I just want to say again, this is no small feat. So there have been studies out there that have looked at studies, clinical trials, and how often is a clinical trial effective, or how often does it work? Less than 10% of the time. So generally between 4 to 6% of the time, you take a drug to trial and it works. And so we had everything against us, and this actually worked, and it did show some benefit in terms of the tumor volume as well as the functional outcomes. So this is really great. And then, of course, I have to say, this is the first medicine that's approved for NF, and so that is also very, very exciting for our population, or for our kids. Where do we go next after this? So it's FDA approved, but there's still some work for us to do. So remember, I told you there was that asymptomatic arm. That was not a part of the FDA approval. So we actually reported the results of that data after the FDA approved this medication. And so I'm hoping that at some point they'll change the indication a little bit, because what we saw is with the asymptomatic patients, those that are at risk for having problems from their tumors, we also saw a response in about 72% of those subjects, and really nobody had symptoms associated with their plexiforms when they were on treatment. And so it truly effectively prevented symptoms in all of the patients. So it would be great to see this ultimately change so that we can better manage some of our patients who are at risk. Adults are also a population that were sort of left out, I guess you could say, of the initial trial. So we stopped at the age of 18. And so the NIH did have a trial looking mostly at adults with the plexiform neurofibromas. And this is just some data and information on that trial. So again, it's greater than 18 years of age now. They got a set dose of 50 milligrams twice daily. But of course, if we needed dose adjustments, then that was possible. Assessments were actually very similar, except for with the adults, we also included a biopsy of the neurofibroma to see how well the drug was getting there, which I think was actually really important data for us to get. And then you had the volume outcomes as well as the functional outcomes, similar to what we did with our children. And you can see on here that the partial response rate was about 62%. So not quite 70%, but still pretty good considering the fact that these patients, the adult patients, have had tumors a lot longer than the kids have. And usually they're not growing quite as fast. And so our thought was that it might not work at all, but it did end up being quite successful. Just to kind of briefly mention some of the other Mech inhibitors, so I focused mostly on the Selumetinib or the Koselugo, which is now FDA approved. There are 3 other ones that are out there that are being looked at. Mirdametinib, or the PD032591 that I had mentioned that we had some good preclinical data for. That one was in a clinical trial through the DoD consortium for patients greater than 16 years of age that showed the data here. So we didn't get quite as good of a response, but we did see some response in patients. And that is now in a clinical trial through SpringWorks, looking at both pediatric and adult patients, which we don't have data on yet, but that study just recently closed for enrollment. We also have Binimetinib, or Mech 162 is the other name for that one. That one is also a DoD consortium study for both pediatric and adult patients, and the response and the toxicity for that, the good data is really still pending, so I don't really have much to share on that study yet. And then Trametinib, of course, is another one that's out there, that there was a phase 1/2 trial for adult and then some pediatric patients. The toxicity for this was very similar to what we saw with the Koselugo or Selumetinib, and the response was a little bit less, but there were a good number of patients who responded to that one as well. So with all of this said, who should go on Mech inhibitor treatment? Because I think that's always a big question. How do you know if you should be on it, or how do you know if your child should be on a Mech inhibitor? And so based on what we know, progressive or growing plexiform neurofibromas definitely respond to this better than those that are stable, and so that would be an indication to go on. Symptomatic tumors definitely would be a reason to go on. So if you're weak or having weakness, difficulty with range of motion, or even pain associated with that plexiform neurofibroma, that could be a reason to start on a Mech inhibitor. And I didn't actually highlight this before, but I should have. Pain improvement with the Mech inhibitors is amazing. You can go from having pain every single day, like debilitating pain, to absolutely no pain. And I've had a couple patients who have had to go on drug holds for whatever reason, and they dread those moments because they know the pain is going to come back. And so it really, really helps with pain. There's also, of course, I would say if the tumor has a potential to cause problems, start a Mech inhibitor. Don't wait for those problems to come. We now know that it works, and I would definitely encourage talking to your doctor about it. And then, of course, right now, the tumor formed during childhood. So if you have a tumor that's growing rapidly that came during adulthood, you might want to make sure it's really a neurofibroma and not something else before you start treatment, because usually it's the ones that formed during childhood that look, the true plexiforms that look benign, that respond to this. Anything that's more aggressive or malignant appearing that comes more in adulthood, you should definitely have worked up before going on the treatment. So what is the future now? So we do have, so we talked a lot about plexiform neurofibromas. There are clinical trials for low-grade gliomas. So remember, I showed you all of the different places that you can potentially get tumors. The action, or the way that these tumors form, is the same, regardless of where it is. So you would think that if you have a plexiform that responds, you should also be able to get a response in a brain tumor. So there are a number of clinical trials looking at Mech inhibitors in brain tumors. Most of them actually are closed to enrollment. The only one that's not is the one on the bottom here, the Selumetinib versus carboplatin vincristine. So this is looking at Mech inhibitor plus standard of care. So this is actually a phase 3 trial, and trying to see which of the two would be better in terms of upfront therapy. And so far, we've actually seen fairly good responses in our low-grade gliomas. So PBTC-029, for example, there was about 9 out of 25 patients that had a partial response, so not quite as good as the plexiforms, but it was a good response. And in the NF stratum, it was probably closer to about 40%. And this is just kind of showing an example of a patient actually that I treated that had the Mech inhibitor. And you can see on the left, this is tumor before and tumor after, so clearly a response from that. Other MEK inhibitor clinical trials, so leukemia is actually something else that you can see very rarely, but sometimes an NF1, same type of an action. And so there is a clinical trial out there looking at trametinib for JMML. And then on here, what's kind of exciting too is for cutaneous neurofibromas, there's now a topical MEK inhibitor, this NFX 179, with hopes that instead of having to take something orally, we can actually treat these cutaneous neurofibromas similar to what we're doing for the plexiforms. So research progress. So I was looking at the questions for my session before starting this, and I actually had this in there already, but I think this is really important because one question asked, my child was on a MEK inhibitor and it didn't work. Why didn't it work? And the answer is we have no idea. And I really wish that we did, but we just don't understand why some patients respond and some don't. But 70% response is amazing, but it's not good enough. We need to get 100% response, and we need to figure out who's best on just MEK inhibitor alone, and what can we do for those patients who don't respond? And so some of what we're looking at now is combination therapy. So what if you take a MEK inhibitor plus what's called a tyrosine kinase inhibitor? So targeting different parts of that pathway, putting it together and seeing if maybe that helps with the response. You can also take a MEK inhibitor plus an mTOR inhibitor. So if you remember in that pathway, there's the two different paths that it can take. If you target both of those paths, maybe that would also help a little bit better. Treatment schedule. So maybe, and this is actually really interesting because in melanoma, they've actually found that you need to have a continuous treatment, but if you have continuous treatment for too long, you actually lose the effect. And so what they're starting to do now in melanoma is they're treating for a period of time, giving a drug holiday, going back on, treating again, giving a drug holiday, and they're finding a much better response with that in terms of the melanoma. So could we do something like that in NF? Maybe. I'm sure a lot of people who've been on these drugs every single day for years and years would appreciate a drug holiday. And so with the PD-032591, the mirdametinib trial, they actually looked at 3 weeks on, 1 week off. But I think we also need to think of other drug schedules. How else can we give this medication so that it gives us the best benefit for the longest period of time? And then the other thing I just put on here is non-tumor impact. And so we know the MEK inhibitor affects the tumor, but what else does it affect in the body, and how can we use that information to help us as well? So we had a few studies that looked at the MEK inhibitors and cognition, and actually showed that it can, when you're on MEK inhibitors, it can improve your the way that you can do certain tests or sort of your executive function. So that's something that's really promising. And I think that we should definitely look into that more with the MEK inhibitors, and especially in terms of treating younger children. Is there a window of time, for example, that we can use this that could actually help that as well? And then I put on here diet metabolic. So this is something that I'm particularly interested in. So remember, there was that one patient that came off treatment because they gained too much weight. There are patients who gain a lot of weight on MEK inhibitors. And so we know that with NF1 loss, there is a shift in your metabolism, or there's some sort of a change in how your body uses energy and nutrition. And so one of the things that I'm trying to look at now through a study at Children's National is how does the MEK inhibitor affect that, and how does it impact the way that you're storing fat, using glucose or other energy sources, and can we actually use that information to identify patients who may or may not respond to MEK inhibitors, but also potentially help with some of these side effects. And then the last thing I'm just going to show, so I in my early days when I did a lot more bench research and a little less clinical time, is I looked at preventative therapy for NF1. And so using a mouse model to try and figure out how can we prevent things from happening. And I already told you that if you have a tumor that's not symptomatic and you treat it right now, we know that it can prevent those symptoms. But what if we started even a little bit earlier? And so we actually had a few studies that we did. The first one was looking at cognition in mice, and we found that if you treat really early on with a MEK inhibitor, we can actually improve the cognitive function of the mice. We then looked at cerebellar function, so motor function, for example, how well mice are able to do the rotor-rotor performances in terms of their strength. And we found that if you treat early on, it improves that. And so then we went next to tumors, and we said, if you were to treat early on, can you prevent tumors? And sure enough, we found that if you treat early in a mouse, you can prevent these optic pathway gliomas from forming. So they don't get the optic pathway tumors at all. And we also did some vision functional studies on our mice, and we found that they had perfect vision, while as when they got the tumors, they had impact on their vision. So very exciting for the mice, of course, but now we have to figure out how can we use this for our patient population. So this is my list of what we don't know, and unfortunately it's a fairly long list. We don't know how long we should treat, when we should stop treatment. Because remember, I showed you if we stop, sometimes it starts growing again. So when do you decide to stop treatment, and how do we know when a good time is? What happens when we stop treatment? Is it going to grow? Is it not going to grow? What kind of side effects are people going to have coming off treatment? When is the pain going to come back? How do we best manage the pain if it comes back? Does this affect fertility? The thought is no, and I can tell you in the mouse model it definitely doesn't, but still that's a question that a lot of people have. Is this going to impact my ability to have children in the future? How can we prevent or avoid side effects? Which patients are most likely to respond, and are there other treatment options that may be better? And so my big take-home points are Mech inhibitors are definitely promising new treatment for patients with NF1 associated tumors. If I have a patient that needs it, I advocate it in every way. They are FDA approved right now, but there are some tumors that they're not FDA approved for, so just keep that in mind. I think that point number 3 is very important. It should be monitored by somebody who knows how to treat with a Mech inhibitor. So it's not something you just go on and then don't go see your doctor for a year. You really do have to be monitored. And then there's, of course, the long-term and short-term effects. We don't know super well yet. So you always have to think about risk versus benefit when you're going to start on treatment. So is this something that you really think would be beneficial? And what is your plan for getting off and things like that? So that's what I have there. I think we have a little bit of time for questions, and I do want to check. There was a couple more questions that had come through beforehand, so I do want to make sure that I also answer those.
They were both pretty specific to side effects. I'm waiting. You're talking, it turns on. It's not that happening. Did it? See, I told you. Okay, so the questions in the app were about some pretty specific side effect things. So gastroparesis and hair loss.
Okay, yes. So gastroparesis, possibly. I would say that we see anywhere from really bad diarrhea to really bad constipation. And so certainly there is effect on the gut. Whether you get the true paresis from it, it really depends on how it's affecting your gut. And so it could definitely be something that you see with that. In terms of the hair loss and hair thinning, so each Mech inhibitor is a little bit different in terms of the side effects. And so I can tell you that there are some Mech inhibitors where we definitely see hair loss and hair thinning. The lighter color hair is also something that we see. So you can go from being a brunette to a true blonde. And so, that is something that will happen, but you can get the thinning with that too. I don't know if whoever asked that, if you want to know which medication is more likely to cause the hair loss, I'm happy to share that afterwards. I just am not sure how much of this I'm able to share to the whole group.
Thank you.
And then the effects on kidneys, there's also a question, does this have effects on the kidneys? So the answer is no, we don't think that this affects the kidneys. And so it shouldn't cause any problems with that. That being said, it can affect your blood pressure. And so it probably is more of the heart side effects than anything else. But certainly if you have issues with your blood pressure, it could potentially affect your kidneys. And if you have issues with other stuff, it could affect your kidneys. And so there's no direct impact on the kidneys that we know of, but there could be sort of secondary effects on it.
Thank you. Yes. Oh, so many questions. I'm sorry, guys. We're going to run out of time.
Thanks for your talk. That's really informative. Yeah. Just curious, is there a significant difference between the side effects seen in adults, like say on selumetinib in this new trial, compared to children, or is there a difference?
Yes, absolutely. Yep. And that's actually, I'm glad that you brought that up because I should have mentioned that as well. So With the young kids, the major side effects that we see are more like skin eczema versus acneiform type rashes. And so you can get really dry skin, you can get eczema with that. I would say with the younger children, I'm more likely to see constipation than diarrhea. And I'm also more likely to see irritability. So I definitely have young kids who go on this who for a little while there are irritable and a little bit hyper, and then it does kind of go away. So that's kind of the younger side. You can definitely see the paronychia in the younger children, a little bit of nausea, but not really actually much. And so in general, younger kids tolerate it fairly well. In the older patients, so older adolescents and the adults, the acneiform rash is really much more prevalent, much more significant. And for some patients really was something that just made them say, I don't want to do this anymore. I mean, I've had a few patients that, you know, despite me starting them on a medication beforehand, despite me putting them on all sorts of different creams and whatever it be, we just could not get that acne under control. And so it was a decision to go off of the treatment. So that's definitely more common in the older adults. Fatigue is more common in the adults that I've seen. I would actually say that diarrhea is more common in adults than what I see in my younger kids. And then the heart issues, I've actually tended to see a little bit more in the adult patients, or the more adult patients than the younger patients. So you can see anything all the way through the lifespan, but adults do have a much harder time, I think, especially initially with this. Yeah.
So are you guys looking into, you know, especially for plexiforms that are more on the on the outside, creams instead of oral?
Yeah, so that one topical Mech inhibitor that I mentioned for the cutaneous neurofibromas, that is a topical cream. Now that's more for the smaller tumors, not the bigger tumors. I think that the big question with that would be how big it is and how much Mech inhibitor you can use topically that, you know, would, because you have to have it absorb, right? And so if you have a huge tumor on the outside and you're putting lots of cream on the outside, it's probably absorbing. And in a way, you almost might as well just take it orally, if that sort of makes sense. So I think it's just a matter of how much you're using on the outside versus the inside. But I actually do think that's a good idea. And these tumors tend to, even the ones that are more superficial, they tend to also be deep. But yeah.
I'm just thinking, I'm like, if I could put a cream on
Yeah.
there and target, if it's the same amount I can do orally, it's
Yeah.
going more targeted.
Yeah, you're less likely to have the acne and stuff like that in theory, as long as it doesn't absorb.
Get into her body, but I'm like I said, you know, we can just, instead of it
Yeah.
going everywhere,
Yeah, goes right there. Yeah.
to that spot.
Yeah. And for people who have a tumor like that, I think that that's actually a really good thought and a really good idea. And the other thing too is that the pain that you get oftentimes with these tumors, you can actually use topical, like for lidocaine, for example, can kind of numb that pain for a little bit. And so the thought would be if you used a topical Mech inhibitor, it might help with the pain similarly to something orally. So I don't know of any groups right now that are looking at topical for plexiforms, but certainly for the cutaneous neurofibromas, they are, and it's something that we in the future may be able to expand to some of these more superficial, smaller tumors.
So,
Yeah. Yeah. Yeah.
so my question is, are you doing clinical studies that check what those long-term effects are, or is that something you need funding, or what kind of following of patients is being done other than just say through AERs? And another quick question I had, I don't know if it's related, is what does it mean when it has a durable effect? Does it get to a point where the tumor just stops and then at least it doesn't grow anymore, or is there always constant improvement? I mean, at least there's pain management, but anyway, two separate questions.
Yeah. So those for the dural response, that one I think will be a little bit faster for me to answer. And really what it is, or the way we define it, is that the tumor doesn't grow. And so you can have your response can be anywhere from 4 months to 42 months after taking treatment. So the maximum response sometimes can take a long time to get to that. But the durable response is once you've gotten to your maximum response, how long can we maintain that without it growing at all? And so that now ties a little bit into your first question where you're asking, are we watching patients long-term? So some of the patients on that phase 1 trial back in 2011 are still on treatment. And so we have good 10 years worth of data on them on treatment, and we're able to see how well they're doing, while they're continuing to take treatment for that extended period of time. And anybody who's on the phase 2 trial who's still on it, of course, we're gathering data from them too. And so the thought would be that eventually, or hopefully, we'll get data that's 10 years out, 15 years old, 20 years out from these patients that we're taking the treatment on these trials. So that's through the trials. Now we also have, though, and that's where the registry that I had mentioned at the beginning is sort of my conflict, that is going to help us get information as well. And so there is a registry for patients who are on Mech inhibitors now through the FDA approval. And so if your child goes on a Mech inhibitor, not through a clinical trial, but just through your insurance now through this FDA approval, you're going to be able to go onto this registry, and the registry is going to collect data on your child from the time they start treatment until the time they stop treatment and beyond. And that's really going to help us get some of that long-term data, which I think is really, really important, because sometimes we see side effects when you're on treatment and we know about those, but we don't know what happens when you stop it. So, I mean, there's a classic example would be a certain type of chemotherapy medication where we knew that it affected the heart a little bit when you were on treatment, but what we didn't know was that 10 years off treatment, it can really affect your heart. And so we don't know that yet. And it doesn't seem like there's going to be a problem at this time, but we have to collect this long-term data. And so I would encourage anybody who has a child who's going to go on Mech inhibitor or is on a Mech inhibitor, as soon as these registry studies open at your site, get on the registry study, because we need to follow kids long-term, and this is one of the best ways to do it.
Excellent. Thank you so much, Dr. Bornhorst. I wish we could keep asking questions. I'm sure there are plenty more, but we'll be starting our closing ceremonies actually in Vevey in about 5 minutes. So can we give Dr. Bornhorst a round of applause?